So here we are going to discuss regulation of gene expression, and in this video we're going to talk about how bacterial genes or prokaryotic genes are regulated. Um, Bacteria divide very fast. They have about 20, for example, in E. coli, it takes only 20 minutes for the bacterial cell to divide and make a copy of itself. So a lot of these metabolic processes have to happen very quickly and efficiently. And as we saw in the previous video, uh, because bacteria do not have organelles, they don't have a nucleus, so transcription and translation are coordinated to happen at the same time. In other words, as the messenger RNA is being made, translation also happens. And bacterial chromosomes are circular and there are no introns. So bacteria have the necessity to respond very quickly to changes in their environment because of how quickly they have to operate. And they have to make sure that they maximize on their energy savings. So, meaning they don't want to waste energy producing nutrients for themselves when the nutrients are available readily in their environment, in which case they have to shut off the genes that produce materials for them. And in the same way, if they have to metabolize something, and if that nutrient is not available, they don't have to keep their metabolic machinery running. So all of these considerations go into how bacteria and prokaryotes generally regulate their metabolic pathways or gene expression pathways. So we already saw one of the ways that bacteria can regulate their the expression of their proteins or the quantity of their proteins. When we were studying enzymes, we saw how in the process of tryptophan production, the final product, which is tryptophan, can go back and inhibit one of the enzymes early on in the series of steps that is required in making the enzyme. Making the product. So this is allosteric inhibition. If you remember, the word allosteric means binding at a site which is not the active site. So this is an example of an allosteric inhibition where a product acts as an inhibitor of the enzymatic pathways that lead to its production, and this is called feedback inhibition because the more product that is produced, the less the pathway will proceed. And the other way of doing this, which is probably actually more efficient, is gene regulation. So here what happens is the entire series of genes or all the genes that are required for the pathway for making the product are shut off so that there is no transcription and you're not wasting any energy in that process or translation therefore. So and there are the way to do this is to turn on a gene or turn off a gene. So here in this discussion, we're going to look specifically at two examples. One is the utilization of lactose and the production of tryptophan. So if tryptophan is present, then the gene in a normal situation, right, when you have when you don't have tryptophan, the cell has the machinery to produce tryptophan. It has all the there are 5 different genes that produce the protein required to synthesize tryptophan. But if the bacteria is in an environment where there is a tryptophan already present, then it doesn't have to waste energy synthesizing new tryptophan, right? So what it does then is it turns off the genes necessary for tryptophan synthesis. On the other hand, lactose is present in the environment, and the bacteria can use the lactose by metabolizing it, catabolizing it to release energy. But if there is no lactose present in the environment, so it doesn't have to spend energy running that machinery, right? So to keep synthesis, to keep breaking down lactose. So in that case, the genes are turned on only when lactose is present. When the lactose is not present in as a baseline, the genes are turned off. So this is turning genes on and turning genes off. So the strategy that bacteria use is by lining up all the necessary genes for a particular metabolic pathway in one continuous stretch. Remember, there are no introns, so it becomes easy. So here we have 5 different genes: A, B, C, D, and E. All trip A, trip B, trip C, trip D, and trip E. All of those are involved in making polypeptides that are necessary for tryptophan synthesis. Together with the promoter sequence and an additional sequence called as an operator, you have all the necessary genes for involved in that particular pathway lined up. Together, all of this, the promoter, the operator, and the genes of the operon, together are called as the trip operon. So operon is simply groups of genes that have related functions grouped together, and we already know what a promoter is. A promoter is the place where RNA polymerase binds and initiates the synthesis of the messenger RNA, transcription of messenger RNA. But what we are seeing here is there is an additional sequence called an operator, which has the ability to bind to a kind of protein called as a repressor protein. And as the name indicates, a repressor is a protein that can bind to the site, and when it does that, so here we have this green region, which is your promoter, which is where your RNA polymerase binds, and then you have your operator sequence, which is present in yellow. And when your repressor protein comes and binds to the operator site, what it does is it prevents the RNA polymerase from moving forward. Therefore, no messenger RNA is going to be made. So here we have an example of how let's go back here. So here we have these 4 genes here in this case, making these 4 different enzymes, and your RNA polymerase, because of transcription factor binding and everything, is going to come and bind to the to the promoter region. But then here you see the repressor binding to the operator site, and therefore the RNA polymerase is blocked. Okay, so a repressor protein turns off genes by blocking RNA polymerase binding to the site and moving forward with transcription. So there are two types of operons. One is called the repressible operon. The other one is inducible operon. Let's take a look at the repressible operon. Repressible operon happens in anabolic pathways. If you remember catabolism, which is catabolism is breaking down of stuff, and anabolism is building up stuff. So here we have tryptophan being built up, created. And to create the tryptophan, we need a series of 5 different polypeptides. When tryptophan is, let me see if there is when there is tryptophan present in the environment, then the synthesis is not required, right? Because it doesn't want to waste energy on synthesizing tryptophan, which tryptophan is already present. So what happens then is tryptophan will come and bind to the repressor protein. In its so at this point, usually repressor in tryptophan is, I think there should be a bit of a picture here. So usually the tryptophan receptor repressor, which is created by a gene which is upstream, which is before where the operon is. So this is a repressor gene, produces a repressor, and that repressor cannot bind to the operator sequence. Okay, so the repressor protein is there, but it is not able to bind to the operator. What that means is the RNA polymerase is going to continue making the messenger RNA here, and therefore make all the necessary proteins necessary for tryptophan synthesis. However, when we have tryptophan present, the tryptophan will go and bind to this inactive repressor protein. And when it does that, the repressor becomes activated. Therefore, tryptophan acts as a co-repressor. It acts with the repressor to make the repressor active. In that activated form, when it is bound to the tryptophan, the activated repressor is going to go and bind to the operator sequence. When it does that, the RNA polymerase is no longer able to bind or continue with RNA synthesis of tryptophan. Therefore, no more new tryptophan synthesis happens. So this kind of operon model is where repressor is activated by the binding of a molecule is called as a repressible operon. Okay, and repressible operon is what we see in we typically see in anabolism, which is synthesis. So here tryptophan is an allosteric regulator of the repressor protein. In an inducible operon, it's the other way around. Here we have the gene usually turned off because the repressor, which is produced by a gene which is upstream. Let me see if there is an image here. Okay, so there is a gene here called Lac So we have here there are three genes involved in the catabolism of lactose: Lac Z, Lac Y, and Lac which correspond which create these different proteins, all part of the lactose catabolism pathway. So there is a gene upstream of this operon called the Lac for the Lac inhibitor, and the Lac inhibitor produces a repressor gene, and that repressor can, in its natural self, can go and bind to the operator and therefore prevent the synthesis of RNA, synthesis of the Lac gene. This is because in a situation where you don't have lactose, you don't want to keep spending energy on the lactose breakdown machinery. However, when lactose comes in, it acts as an inducer. It goes and binds to the repressor protein, and when it binds to the repressor protein cannot bind to the operator anymore. Okay, so it is actually inducing the synthesis of the lactase genes. So the binding of the inducer, which is a version of lactose, a steric form of lactose, allolactose binding to the repressor is going to make the repressor inactive, which means it is no longer able to bind to the operator site, which means that now the RNA polymerase is free to go ahead and make more messenger RNA. And this is just a quick additional note. Things can be a little bit more complex than a direct inhibition and repression. There can be like little bits of tweaking up of the system depending on the situation. So here we are seeing a situation like this. Bacterial cells would prefer glucose to lactose any day. So when you have glucose, it doesn't want to use the lactose operon. So when you have a lot of glucose, then you have let's first look at the situation. When you have less glucose and you have more lactose, you want the lactose operon pathway to be active. So in addition to the to the promoter site and the Lac site, there is another site here called the cap binding site. And when you have a lot of lactose and very little glucose, you have more of this molecule called cyclic AMP. And cyclic AMP is going to bind to an inactive protein called cap protein, making it an active cap. And the active cap protein complex is going to go and bind to the cap binding site, which causes more and more mRNA synthesis. Okay, so I'll repeat that. When you have more lactose and less glucose, you have more cyclic AMP, which goes and binds to an inactive cap protein, making it active, which will go and bind to the cap site, which is next to the promoter region, and therefore make more and more messenger RNA. This is basically saying let's catabolize more lactose, because more lactose is present, less glucose is present. But when you have more glucose, that means it doesn't matter whether lactose is present or not. It wants to use glucose better than it wants to use lactose. So what it does is, when more glucose is present, cyclic AMP levels go down. Okay, when cyclic AMP levels go down, then the cap protein, inactive cap protein, is not bound by anything, which means it cannot bind to the cap site, which means that the inhibitor, which means the production of lactose enzymes is going to be reduced. Like lactose operon is going to be shut down, not shut down, but reduced. So that is an additional level of control. When you have a site such as a cap binding site which responds to another metabolite, here we have glucose, which is regulating the level of cyclic AMP, which in turn is regulating binding to another protein, which is a cap protein, and that regulates how much of messenger RNA is being made or not made. So, in summary, we have two types of operons. Operons are sequences of genes that are all performing the same function clumped together, and there are two types of operons in general. One is a repressible operon, which is involved in anabolic pathways, and we have an example of that is the tryptophan operon, and then we have inducible operon. Example of that is lactose operon, that is involved in catabolic pathways. And in both of these cases, the objective is not to waste energy. It is trying to not produce proteins when it shouldn't be making proteins, so that the cell can allocate that resource, that energy, for other purposes. So hopefully this was helpful. It might seem a little bit complex from the outside, but just look through this a couple of times and I think you will realize that it's very straightforward. It is how it's whether the repressor can bind to the controller, to the operator site in its baseline state or not, and how the binding of another molecule is going to either induce it or repress it. So as long as you become comfortable with that, this area of regulation of metabolism, or regulation of gene expression in prokaryotes becomes quite easy to understand. Hopefully this was helpful. Thanks.